So I have the pleasure of introducing you to Samhita Yadavalli. She is a more recent addition to the Waksman Institute at Rutgers, and her research focuses on how bacteria respond and adapt to antimicrobial stress. Dr. Yadavalli is particularly interested in the roles of small proteins in these adaptations. Before being appointed at Rutgers, Dr. Yadavalli was a postdoctoral trainee at Penn and earned her PhD at Ohio State. In her short career thus far, she has mentored several graduate, undergraduate, and visiting scientists, and even a high school student. And you can see on the screen, her talk is entitled "An Epitranscriptomic Enzyme Moonlights in the Bacterial Stress Response." Samhita.
Can everyone hear me okay? Great. I'm really thrilled to be presenting here at this Waksman Symposium, celebrating Dr. Witkin's 100th birthday and her amazing achievements. Dr. Witkin, happy birthday, belated happy birthday. And you're truly an inspiration for all of us. I'm so excited to be in the same building as you've been. And I know everyone's eager to get to hear the guest of honor. So I'll just jump in and try to keep this short. Joanne, please feel free to jump in if I'm going over time. All right, so so I guess before I start, I want to say, like Dr. Witkin, I'm also in love with filamenting snake-like E. coli. So my lab studies bacterial stress responses to different kinds of signals, including antimicrobial compounds, low magnesium, low pH, and we are also studying small protein regulators. Today, I'm going to talk to you about a story where short rods of E. coli cells that typically look like on what I'm showing on the left side panel grow into these beautiful long filaments when exposed to antimicrobial peptides. And I want to make it clear that this is not due to the direct action of the antimicrobial peptide on the cell envelope or some damage caused by it. Instead, these peptides are sensed by a signaling system, which then leads to this block in cell division by a mechanism that was not really straightforward to decipher, as I will tell you about. But why do we care about filamenting E. coli, right? I mean, in most cases, it's a stress response. And for decades, filamentous cells have been considered sick or dying members of the population, which is true of many mutants of cell division proteins. However, fermentation can also be a survival strategy in lethal environments. And I'll come back to this point later on in my talk. So as an outline, I want to give a brief introduction to stress response in bacteria. Then I'll talk about the antimicrobial peptide stress response in E. coli and talk about some of the recent advances we've made in understanding how a key player in this pathway functions, and then summarize the talk. So as we all know, bacteria have to face constant challenges in their environment, including antimicrobial compounds and changes in temperature, pH, etc. And in E. coli alone, we have more than 30 signaling pathways that help respond to such signals. And in general, signal transduction is predominantly carried out by two component systems. And the first component of these systems is a sensor or a histidine kinase, typically, which detects the environmental signal, and then it's passed on to a response regulator. The response regulator is typically a transcription factor that modulates downstream gene expression and helps the organism adapt and survive. So how do E. coli respond to cationic antimicrobial peptides? As I was studying this, one of the first observations I made was E. coli cells filamented when they were treated with sub-MIC levels of cationic antimicrobial peptides such as C18G. Now, C18G is a derivative of a human platelet factor. And what I noticed was under sub-MIC levels of this peptide, the cells grew as really long filaments. These were hundreds of microns in length. They varied a lot from really short to there was a lot of heterogeneity. And when you don't have this peptide, the cells grew as you would expect as short rods. This peptide has been shown to activate the FOCI signaling system. And the FOCI two-component system is an important regulator for hundreds of genes. And I've mentioned that it's also important for magnesium homeostasis and other processes that I've listed here. So one important detail about this pathway is that there is a small protein called MGRB that is directly regulated by this two-component system, FOCI, mediating a negative feedback loop right here. And this, the protein is important because inactivation of this protein or its interaction with FOCI, this inhibitory signal here, leads to acquired resistance to colicin in clinical isolates of Klebsiella pneumoniae. So what we had noticed in case of MGRB deletion is that, can everyone see this screen now? Okay, great. So in just like we saw in cells that were treated with antimicrobial peptide C18G, in cells that were deleted for MGRB and grown in a FOCI activating condition, we see that the cells are really, really long in filaments, and this phenotype is suppressed by deletion of FOCI. Can you see the delta FOCI on the screen? Great. Okay, so as we can see, the filaments are really heterogeneous and the sizes vary from tens to hundreds of microns. Sometimes it's really hard to even image them because they're spanning many fields of view. And so one of the important things about these cells were the filamenting cells did not show an obvious growth defect relative to the non-filamenting cells. And the phenotype is completely reversible. By that, if you remove the peptide or if you grew these MGRB deleted cells in non-FOCI activating conditions, the filaments would become short cells again, and they're just fine. So what was known and what we understand is that the FOCI sensor kinase is de-repressed in these cells. So one of the things that we looked for is whether there is upregulation of this pathway and if this FOCI system is highly activated under these conditions. And indeed, in both these conditions where we see filamentation, it's FOCI dependent and the cells are strongly activated in this pathway. So we wanted to characterize these filaments better, and we did a membrane stain to look at whether these cells have any septa or partitioning, and we did not see any. And in addition, we were able to show that they have a continuous cytoplasm. So what is the mechanism of cell division inhibition in these cells? One of the mechanisms that we looked at was the SOS response, right? So this audience, more than anyone else, is very familiar with that. So one of the first steps in the cell division of E. coli is the recruitment of the protein FTSZ to the division site. And FTSZ is a prokaryotic homologue of tubulin. And when there is DNA damage, the SOS response kicks in and the cell A protein, which is induced, goes and binds to the FTSZ monomers in this overly, as shown in this overly simplified scheme, and it inhibits the Z ring from forming. And what we noticed in these filaments is that the nuclei were well separated and they were nice and multinucleated. So there was no problem at least visually at the level of DNA segregation. And we did not see any suppression of this phenotype in a delta cell A mutant. And more directly, we were able to show that the Z rings are formed intact in both the filamenting and non-filamenting cells, which suggested that there is a defect in cell division somewhere in the later stages of after the Z ring formation. So what I've shown you is that high activation of this pathway, FOCI, leads to increase in the FOCI regulated gene expression and somehow leads to the cell division defect. To understand which are the other proteins that are part of this pathway, we did a genetic screen, a transposon insertion mutagenesis screen for suppressors. Now, if you remember, I was just telling you that there is no screenable obvious phenotype for limiting cells, but what we were able to do is, after a lot of trial and error, find a specific agarose that has really low trace amounts of divalent cations that was suitable for such a screen. And without going into too much details, one of the complete suppressors we got was in a gene called QueE. Now, when we made a clean deletion, we were able to see that this phenotype was suppressed in both the conditions where we see filamentation. And we were able to restore filamentation by plasmid complementation of QueE. So what is QueE? This is a protein that participates in a quercetin tRNA modification pathway. And quercetin, or Q, is a hyper-modified guanosine, universally found in specific tRNAs. It's also found in humans, but most of the eukaryotes use free QueE base from either gut bacteria or diet. And this modification is really important for translation fidelity. And as shown below, this pathway starts with the precursor GTP, and QueE participates in one of the initial steps leading to a key intermediate formation, which is called pre-Q0. And after various other steps, the final product qtRNA is formed. And we wondered if this pathway is linked to the filamentation phenotype we're seeing. We did a lot of detailed genetic analysis, and we're disappointed to find that actually it's not linked to the filamentation phenotype we're seeing. One thing we didn't know was if we express QueE on a plasmid, the E. coli cells are filamenting from this overexpression. So we hypothesized that QueE expression is upregulated in these filamenting cells, and we directly checked that by making transcriptional fusions of QueE-YFP. And I'm just showing the data for C18G here. So in the presence of the antimicrobial peptide, the FOQ system is turned on, and indeed QueE expression is much higher compared to a condition where there's no activation of this pathway. So the next thing we wanted to see was, how is the upregulation of QueE responsible for inhibition of cell division? Is QueE directly or indirectly affecting this process? So for this, I want to just quickly show you a schematic of the cell division pathway in E. coli. As you can see here, there are a lot of steps involved and a lot of important proteins in this pathway that are essential. So one of the things that our previous screen wouldn't have picked up is essential genes, because we are doing a transposon insertion. So we went ahead and did a spontaneous suppressor screen by overexpressing QueE with the goal of finding any proteins that are downstream of QueE in this pathway. And indeed, we found mutations that suppress this phenotype in both FDSZ and FDSA proteins. Now, this is sort of gave us the clue that QueE might directly be interacting with the divisome. So basically, we tested this by localizing QueE. We co-expressed a live fusion of QueE along with an M-cherry fusion of FDSZ to localize these two proteins in these filamenting cells. And we did indeed see that QueE co-localizes with the Z ring. This was really exciting. So basically, we found a new function for QueE under these conditions of antimicrobial stress response. So in summary of this model, under low stimulation of the FOQ-FOP system, there is basal transcription of QueE, which leads to sufficient amounts of QueE being produced for its tRNA modification function. But when there is a high stimulation of this pathway, we see that there is upregulation of the levels of QueE, which is not only sufficient for its tRNA modification function, but also the excess goes and binds to the division proteins and inhibits cell division. So some of the questions that we're really interested in following up with are whether these two roles of QueE are functionally independent, and what does QueE interact with in the cell division complex, and whether this type of filamentation has a growth or fitness advantage under the stress condition, and what are the levels, what are the effects of these upregulated levels of QueE on translation in the cell. So these are questions that we are currently exploring, and recently in the past year, one of the graduate students who just started working in my lab has been working on answering some of these questions. So one of the first things he set out to answer is whether these two roles of QueE are independent. So as I've shown you, these two phenotypes are very distinct, and we needed to have separate readouts to be able to assess these two activities. So as a readout for its cell division inhibition function, we use microscopy to look at the morphology of the cells. And then for qtRNA formation, we used an assay, which is a modified northern blot assay, using a chemical that can cause slow migration of the modified tRNAs relative to the unmodified tRNAs denoted by G over here. So using these two assays, we basically first wanted to see if there are other enzymes out there that are different from the E. coli enzyme. So interestingly, in the literature, there's a lot of work done on biochemical characterization of the B. subtilis ortholog QueE. And this particular enzyme has only about 19% similarity or 40% homology with that of the E. coli enzyme, showing that there are a lot of rest of the sequence that's really divergent from this E. coli counterpart. So one thing that we wanted to check was whether the Bacillus subtilis ortholog is able to cause the same kind of inhibition of cell division. And when we looked at that, we found that the Bacillus enzyme is not able to cause filamentation, whereas it is fully functional in forming qtRNAs, as shown in the northern blot on the right. Now, we also checked some of the more closer relatives from Klebsiella and Salmonella, which are both able to cause filamentation in E. coli, just like the E. coli QueE, and are also functional in forming qtRNAs. But this data basically suggested to us that these two functions can be decoupled. And we were really interested in finding specific residues that could be responsible for uncoupling these two activities. So we took a look at the recently published structure of the E. coli QueE from 2019, and we identified a set of residues that are really important for its function. And as shown in the table on the right, these residues are all important for various aspects of the catalysis of qtRNA formation, including substrate binding, ion sulfur clusters, ion sulfur cluster binding, and acetyl methionine binding, and also for acetylation of this protein. And then we basically, Samuel basically, my student, did an alanine scanning experiment where he made single mutants of each of these residues. And we were able to, again, put them through the two assays that we've been using to characterize these mutants. And what we saw is we were able to isolate residues that affect only qtRNA synthesis. So one example is the R27A mutant, where the QueE variant is able to still cause filamentation or cell division block, but is unable to form modified qtRNA. And then we found residues that were affecting filamentation only. So the examples include the C31, C35, and K66 residues that are unable to cause cell division inhibition, but fully functional in qtRNA formation. So we also found some residues that did not affect neither of these functions, that did not affect either of these functions, and also affected both the functions in case they caused misfolding of the protein. So basically, this is all part of ongoing research right now, and we are in the process of figuring out more details of this moonlighting protein and its function in the antimicrobial peptide stress response. And in summary, what I've shown you is that the filamentation phenotype is not the result of the direct action of the antimicrobial peptide on cell envelope. Instead, it is due to the block caused by strong activation of the 4Q4P system. And cell division is inhibited post-Z-ring formation by this enzyme called QueE, which could be an auxiliary cell division checkpoint under these stress conditions. And QueE's function in tRNA modification is independent of its function in cell division. And it's an example of how epitranscriptomic enzymes can potentially act as regulatory links between translation and stress response. And I want to leave you with this final thought where filamentation can act as a survival mechanism in bacteria. And some of the work from UPEC by Cheryl Justice's lab has shown that UPEC bacteria, in case of IBDs and other physiologically relevant situations, are able to evade by filamenting. They're able to evade phagocytosis better than cells that are unable to form these filaments. And so it's a really interesting concept, and we are studying this further in our context. With that, I'd like to thank the people responsible for the work. A lot of the work I've shown you in the beginning, it's from Mark Goulian's lab, and members of Goulian lab who have contributed directly to this project. And currently, Samuel Adelaide, a talented student in my lab, is working on some of the questions that I talked to you about. With that, I would like to thank everyone and take any questions.
I just wanted to point out that when Sam said that she's honored to be in the same building with that had previously housed Dr. Witkin, she could actually have moved one step closer. She is occupying the same laboratory within that building as Dr. Witkin.
That's wonderful.
Oh my God, thank you for adding that, Richard, because I really did not have any idea about that. That's a great honor.